Okay, the previous video gave an overview of the cellular respiration process where glucose is oxidized to form carbon dioxide and oxygen gas is reduced to form water. And in the process we make lots of ATP energy. Now this happens sequentially over a number of phases, and those 4 phases we want to introduce to you now. Glycolysis is the first, where glucose is broken in half to form a molecule called pyruvate. Next is the preparatory reaction, where pyruvate is modified to enter the citric acid cycle, which is the third step of the process, where the remaining carbons from the original glucose molecule are completely oxidized to form carbon dioxide. And then finally the last step, the electron transport chain, where we make the bulk of our ATP out of this process using energy from electrons that have been gathered. Okay, so let's start with glucose outside the cell, and it's going to diffuse with the help of a transport protein down its concentration gradient to inside the cell. Now once it's in the cytoplasm of the cell, glycolysis will happen, which breaks those glucose molecules in half. And in the process we gain a little ATP. So let's take a closer look at glycolysis. Okay, the first step of cellular respiration is glycolysis, which literally means the splitting of glucose. And this occurs outside the mitochondria in the cytosol surrounding that organelle. So here we see that 6-carbon chain of glucose is split directly in half to form 2 3-carbon molecules called pyruvate. Now in the process of doing that, we will gain 2 ATP as part of this step. But so far we've just broken the glucose molecule in half. Now those molecules of pyruvate will then be able to move into the mitochondria, specifically the matrix of the mitochondria, the very inner space. That's where we're going next. Okay, so here you can see our 2 molecules of pyruvate inside the matrix of the mitochondria. Recall the cristae, that inner membrane that folds around inside the mitochondria, you can see there. Okay, so this next step, the preparatory reaction, happens inside the matrix of the mitochondria, and we're going to see pyruvate, each of these pyruvate molecules, lose one of their carbons, forming CO2. And of course that CO2 leaves through simple diffusion, and that's some of the CO2 that we breathe out. Now that prepares these molecules to enter the citric acid cycle in this 2-carbon form. Okay, so that third step or phase of cellular respiration is the citric acid cycle. This also occurs in the matrix of the mitochondria. And these remaining carbons are all oxidized to CO2. So think of those original glucose carbons getting finally turned all into CO2, and again through simple diffusion leaving the cell to be respired out. In the process we'll make a couple more ATP. However, what's been happening this whole time up through all of these steps is that we've been scavenging some electrons from each of these reactions, and these electrons have been sort of put away, if you will, into these molecules that are called NADH and FADH2. We've been saving them up, and now those molecules are going to move to the cristae, that membrane, for the last step, the electron transport chain. Okay, so here's the electron transport chain. Recall this membrane is the cristae, which is inside, it's the inner membrane of the mitochondria. What we see here are a number of proteins spanning the membrane. Now what happens in the electron transport chain is that these electrons we've been gathering and sort of saving as NADH and FADH2 are going to be transferred from one protein to another, thus the idea of transporting electrons across a chain of proteins. In the process, that is going to pump these hydrogen ions outside into the intermembrane space between the 2 membranes of the mitochondria. So what you see here is this pumping action. We're using the energy from the electron being transferred to pump this hydrogen out. Now what's pulling this electron the entire time is the presence of the O2 molecule down here. And you can see that what happens when that electron is finally met by O2, it is reduced, oxygen is reduced to form water. And it's that pull on that electron that keeps this whole process going, which is why respiration requires oxygen. Now what happens over time is that a number of these protons, of course, are built up outside, and we now have a concentration gradient. And this molecule here, ATP synthase, which you can tell from the name is an enzyme, will use that gradient to produce ATP. It will use the potential energy caused by the gradient to make the ATP molecule, and it will continue to shuttle those back in, making lots of ATP in the process. Okay, so what you should do is pause for a moment and ask yourself which step makes ATP, which makes CO2. Some steps make both, and see if you can go through this and quiz yourself to remember the important parts of each of the steps. Okay, so glycolysis, the first step, will make 2 ATP, but remember it's just breaking glucose in half. No CO2 are produced during that step. The preparatory reaction does not generate any ATP, but we did make a couple of CO2 molecules. The citric acid cycle, a couple of ATP were made during this process, and the remaining carbons from that original 6-carbon glucose molecule have been oxidized to CO2. And then finally, there's no more carbons to oxidize to CO2, so no CO2 is produced, and that electron transport chain generates 34 ATP from all of the electrons that have been gathered through the process. Thus, one glucose molecule can create 38 ATP molecules for energetic processes inside the cell.